Okay, well, ladies and gentlemen, welcome to the Computer Science Colloquium. Next week we will hear from Lala Titakola, who is the Chief Operating Officer of Wikimedia. So if you are at all interested in Wikipedia or Wikimedia, next week should be a must for you to attend that. Today I have the pleasure of introducing you to Tom Slezak from the Lawrence Livermore Laboratories. He's going to explain why is it that we don't have an Ebola vaccine yet. Let's welcome our speaker.
Thank you very much. It's really a pleasure to be here. This is actually the second time I've been here. Dr. Ledeen had me here about 9 years ago to give a talk. I should, in terms of full disclosure, 40 years ago I was a student of Dr. Ledeen at USF, so we go back a long ways. There's a lot of things I'd like to talk about, not a whole lot of time, so I'll go through some of them quickly. I'm happy to take questions in the middle, especially if I say something that you don't understand or use terminology. Otherwise, we'll try to get some time for a few at the end. The idea here, I want to talk a little bit about bioinformatics in the context of this question of, there's been tremendous advances over the last couple of decade and a half at least, but it's not translating yet into what you see when you go to your health clinic. And I'll talk a little bit about why that's the case. So I'm part of a biosecurity program at Livermore. I've actually been there a long time. I've been there 36 years full-time. I was also there as a summer student 40 years ago this last summer, and I got my master's in computer science through UC Davis there. I've worked the entire time as a computer scientist in the various areas of bioinformatics. I was part of the proto-beginnings of the human genome program, which actually we started up work on that 30 years ago this summer, 1984. Worked on that for about 14 years. I got really sick of the politics, and I very naively and very stupidly thought there'd be less politics in biodefense than there was in the human genome program. So in about 2000, started building up a pathogen bioinformatics team, and I'll talk about some of the things that we've done. I'm just going to talk about our detection and diagnostic work. We also do a lot of work in decision support, both classified and unclassified, medical countermeasure design. There's a lot of capabilities we have. I'm mostly just going to focus on the bioinformatics. Livermore, we always like to show you these pictures of these $90 million supercomputers, but hey, the physicists own those, and we do not get any time on them for biology. We make do with a lot smaller machines. So I have a pretty large team. This is a bunch of the people. It's about a 2-year-old picture. It's a multidisciplinary team. This is the largest team I'm aware of in the country and in the world that is solely focused on pathogen bioinformatics. So we worry about how to do more efficient detection of pathogens, and that's basically it. We started out working only in biodefense lately. We've been segueing that into regular public health. The events going on now in West Africa and indeed worldwide with Ebola really bring to the forefront the kind of work that we've been working on for over 15 years. And I'll tie some of that in as we go along. This team is composed about half of people have biology degrees, and the rest of us are computer scientists, some PhD bioinformaticians now, PhD biostatisticians. It's a really great team. These are a fun group of people. I'm kind of like Dr. Ledeen. I could retire any time, but hey, I get to work with some of the brightest people in the world, so what would I be doing if I wasn't coming in and doing that? We've done a lot of things that are a little bit more than just sit at the keyboards and pound away. We actually get our stuff out there. This is a photo I took on October 4th or 5th, 2001. It was the early in the morning after the first anthrax death. We got a call at Livermore. We'd been building a system for the 2002 Winter Olympics in Salt Lake City to do wide area biodetection. We got mobilized and awakened in the middle of the night, 2 in the morning, told to be at work at 4 in the morning. They wouldn't tell us where we were going or how long we were going to be gone. Put us in our gear on a C-130 and we flew. That's the longest, slowest ride across country I've ever had to DC area and set up our system and started running it. And that was kind of how we got out there. These are some shots from what it looks like in the lab we're running. This was actually the 9/11 one-year memorial in this is Times Square. It's one of the aerosol detection units. Air sucked onto filters. The filters are taken into laboratories where PCR signatures are run looking for the presence of biothreat agents. The general idea here is if somebody takes a crop duster and flies over Manhattan or San Francisco or wherever and dumps kilograms of anthrax, plague, things like that, you'd like to have an early warning. You don't want to wait 2, 3, 4 days till all of a sudden tens or hundreds of thousands of people start getting very sick and showing up in the emergency room. So this is kind of an interesting intersection. We are not a research group per se. We're a deliverable group. We build things that people ask us to build. Occasionally we get a chance to publish a little. So one slide of Biology 101 for those of us computer scientists. I had exactly one biology class in college, so I've had to pick it all up on the fly myself. But we worry about bacteria, single-cell organisms, viruses, nucleic acid and protein, sort of this on the edge of life, and things like Ebola certainly are viruses. We also worry about fungi, yeast, and various parasites. Toxins are proteins that are produced by some pathogens. So this is the realm of what we worry about. We use DNA detection techniques mostly for these. Since these are proteins, we have to switch and use various protein detection techniques. Sometimes we also look for the DNA of the organisms that contain the genes that create the toxins. This is really what we do. We look at lots and lots of sequence. As computer scientists, the big challenge here has been scaling. And this has just been a wild ride for me for the last 15 years. When we started doing this, there were 62 genomes of bacteria in the public database. 62, that's it. I used to joke that it was a Noah's Ark for singles. There was one anthrax genome, one plague, one so forth. Now there's just hundreds of thousands of genomes. The explosion of data caused by the cheap next generation DNA sequencing has just been phenomenal. So every algorithm that we wrote in the last decade broke completely 3-4 years ago. We've had to work constantly scaling. It's a full employment act for computer scientists, the way biology is moving. So this is kind of a busy slide, but the issue here is, why is this a tough problem? A couple of reasons. There's a lot of natural variation, a lot of different strains. Some of those strains will kill you, some of those strains won't kill you. They might kill your dog, they might kill an aardvark or whatever, but trying to sort out what's deadly to humans versus other things is a big problem. A lot of genetic near neighbors, things that have DNA very, very close, maybe 99% or even higher in common that aren't deadly. We have to sort those out accurately. The RNA viruses, and these are things like Ebola, they have very high mutation rates. They do not have the DNA repair genes that are in the DNA organisms. Therefore, their mutation rates are 1,000 or more higher. So these things are just constantly kicking off different strain variations, some of which may survive, most of which are not going to do anything. Some of them may be more deadly, some of them may skip hosts. So for example, things like with HIV, at some point, some variant made the jump from non-human primates to humans. So these are things that we have to really worry about. If you do have a detection for the Ebola of today, there's nothing that says that the next outbreak that happens 2 years from now in the Congo won't evade your detection reagents because of this constant change. So it's a very, very interesting work. The first thing we need to do when there's an outbreak is somebody quick get some sequence so we can understand what's going on. Also, these can be very harmful to you or deadly at extremely low levels. The current Ebola outbreak, they're saying it's not even worth looking for the Ebola virus using sensitive PCR until you have a fever of at least something like 101 degrees. They keep changing the number. So these things can be in your body, they can be preparing to do great harm, but you can't see them until it's almost too late in some cases. And of course, we have to worry about new, emerging, and certainly genetically engineered organisms. The field of genetic engineering has been changing incredibly fast, most all of it for good uses, depending on where you stand on GMOs and things like that. But it is now the case where you don't need a state-sponsored program to do really deadly genetic engineering. An angry graduate student in a good university could cause a lot of problems. And finally, the biggest problem of all is very complex backgrounds, whether it's in the human sample or in the environment. You're trying to find, it's not a needle in a haystack, it's a grain of sand on a giant beach. It's really a very difficult problem. So over the years, we've gotten to be really good at detecting known biological threats. And we live in this sort of a space. On one axis, there's time from sample in to answer out, and then there's cost. And we can look at how the known, emerging, and advanced pathogens play out against different techniques. PCR is a pretty simple technique. I don't have time to go into it here, but you've got two bits of DNA, one on a forward strand, one on the reverse strand, and it allows you to amplify up whatever lies between them. It's very accurate. This is the same technique that you see all the time on the CSI type TV shows. A variant of it is used for human identification, and we use a different variant of the same technique for identifying the pathogens. It allows you to get billion-fold amplification of just the thing that you want in the sample. So it's sort of like saying that one grain of sand you want to find on a giant beach, it now turns into a house-sized block, and it's real easy for you to identify. Microarrays, I'll go into in detail. This is something we've done a lot in the last 7 years. Everybody knows about DNA sequencing. If it's a brand new outbreak, it's killing people, it's a new strain of Ebola, and the CDC's detection reagents don't work, your only hope is to go in and sequence it, find out what's going on, create new reagents. So we work in this space, and we have to tailor what we do to the particular problems at hand. So let's go back to this context of pathogen detection technologies and what's going to happen in the future. So hypothetical question, what are we going to see? Are we going to see smartphone-based personal health monitoring? Are we going to see point-of-care diagnostics in the health clinic? So a drop of blood gets taken and you get the answer quickly. Do you have Ebola or whatever? Not. How about symptomatic multiplex PCR panels? So if I come in and I've got something in my lungs, is it, what kind of bronchitis or pneumonia is it? What's the organism that's causing it? What about DNA sequencing? There are some people who will say that every single sample is going to be sequenced, and all these other technologies are obsolete. We're going to do everything with sequencing. I'll drill down on that a little later. And then there's this personalized medicine. So each of us has a unique genome ourselves. And so if a particular drug or a vaccine comes out and we all take it, most of us will be fine. One or two of you might have a bad day, and maybe one or two people in 1,000 or 1,000,000 or 10 million might have their last day. So how do we work out the personal variations in your own genome with what the pathogens will do to you and what the drugs that we give will do to you in the presence of that pathogen? So the short answer is all of the above. All of the above are going to happen. This decade is going to be the most interesting decade in terms of medicine and science that's happened yet. The rest of this decade, we're going to see some pretty amazing stuff. So those of you who are students here, you're coming out at just the right time. It's going to be fascinating. So just a few things on some of these techniques. So we've done a lot of work over the last 6, 7, 8 years in large multiplex PCR. The kinds of things we've done, 28 signatures, a 28-plex biothreat surveillance panel. This actually was in use in about half of the US cities that are doing this daily check for pathogens. This is actually called the BioWatch program. Has anybody here ever heard of the BioWatch program? Probably not. It's one of the most successful things that nobody knows about. Over 75% of the US population is under the BioWatch umbrella, meaning if somebody does fly a crop duster over a city and sprays out a few kilograms of anthrax or something, it will be detected within 24 hours, and at least we can then start dealing with it. You're a little bit too far out from under the umbrella here in Sonoma, sad to say, but if you're down around the Bay Area, you're covered. We've also done a foot and mouth disease one. Foot and mouth disease is an interesting disease. It doesn't kill people. It actually doesn't kill the animals. But what it does kill is international trade. So if I were a terrorist and I understood Americans better, I wouldn't be trying to make people sick. I would just go in and release some foot and mouth. It's really easy. It's endemic in all the areas where the terrorists are. It would absolutely cripple our cattle, pork, and sheep industry. It would cut off all exports. People would be freaking out. McDonald's stock prices would crash. You guys wouldn't know what to eat for dinner. It would just cause absolute economic terrorism, which is actually more effective than blowing up people or making a few people sick and dying. So this is something that's kind of interesting. There's also, we did an avian influenza one. There's been politics behind all of these. All I'll say is these panels were in half the cities, and somebody at CDC, a new guy came in and said, oh, I don't like how these were validated, so let's throw them out. We had these running in all 14 of the national animal health labs around the country, including one in Davis. And the labs loved them, and then somebody at USDA said, we didn't invent these, so throw it out. This one, we were told, well, we were being funded by DHS, and DHS can't worry about humans, so that's why it says avian. We were told we could only study influenza that affects birds. Well, fortunately, the same bird flus that we worry about as humans are also for birds, so it was a stupid thing. But this is all politics is science and all science is politics is unfortunately true. It'd be real nice to have an Ebola rule-out panel now. If you follow what's going on in West Africa, you'll notice that there's some pretty tragic situations. They can't really identify what people have. People come in with a fever now, chuck them into the Ebola ward. We've seen some things that show as many as 60% of the people who are coming in with a fever do not have Ebola. Probably about 100% of them do after a few days in the Ebola ward, though. So they don't have the ability to sort out, is it malaria, is it typhoid, is it any one of a whole lot of other pretty interesting things that you could get in a jungle environment in West Africa. I have seen no fewer than 4 calls for proposals in the last 2 weeks for multiplex diagnostics that include Ebola from different agencies. And these are things that they should have been doing years ago. But George and I were talking on the way over here, we only react to things, we don't really think about it in advance. Switch gears here, we also have been doing work in sequence analysis. At Livermore, we've been watching the rate of development of sequencing technology, and so I made the call about 7 years ago that we were not going to buy a sequencer because as soon as you've got it in and got it up and running, it's obsolete. Instead, I just call on my friends in universities and other labs who have a whole bunch of boat anchor sequencers, or soon-to-be boat anchors, and have them do the sequencing for me. So we outsource it. But what we have focused on is efficient analysis of the data. So this is looking at the problem of what's called metagenomic sequencing, which just means a dirty sample. It's not a pure culture scraped off a culture plate. You've all seen those petri dishes with the little anthrax cultures growing on them. That's the easy sequencing. The hard sequencing is somebody takes a cotton swab and runs it along a surface or a baseboard in a building and then says, is this a facility where they're doing something with anthrax or other deadly things? You're looking for, again, very small amounts of something in a huge background of whatever the crud is that's on every surface that we touch. So what we've done is try to figure out how to solve this problem in a way that would allow sequencing to be done anywhere people want to do it. And I can't name things like who sponsored this work or any details, but this is a 4U Linux box server. It's got a terabyte of RAM, 40 cores, up to 16 terabyte of disk, depending on how much we put in there. We use SSDs usually. And this allows it to be taken anywhere with a sequencer and get answers in a few hours. I got a number from a colleague at a federal agency that if somebody is to fly a sample back, so there's a rush sample somewhere in the world, some hot zone in the world, it costs about $500,000 to get a sample transported back to one of the US labs in the United States. And about 80% of the time the samples got cooked along the way because the dry ice ran out or somebody didn't refill it. So this is what we've been working on, is trying to allow microbial forensics to happen quickly at the point of need. There's a lot of other things I could say, but since this is going to be on the YouTube, I will not say any more on that. So what we've done is we've built some very scalable software. Any of you who are interested in it, just do a Google search on LMAT and Livermore. And this is an open source tool. It is extremely fast and extremely thorough. So we can get down to the strain level across the bacteria, viruses, and even the larger organisms. We have a whole lot more data because we can also use the draft, the unfinished genome data. So this, again, was one of the things we did for a scaling challenge. I had my team start this work about 5 years ago. I noticed everybody else was throwing cores at the problem. I had friends that said, I've got a 2,000 core machine doing these genome analysis. Another guy, I've got 20,000. I said, well, let's go along the other axis. What is the best data structure you could build? So if you had an infinite amount of memory and you didn't have to worry about it, how much information could you squeeze out of this data? And so that's what we did. Besides the DRAM on here, we also have been doing a lot of work with NVRAM. So basically, flash drives, a little more expensive than USB sticks, and in some cases up to like 20 terabytes of that. So I think this is a kind of an important thing for those of you as computer scientists is don't get stuck just worrying about one axis. Don't just think about computation power. Think about things like memory. And think about what's the sweet spot for what you're trying to do. It's been quite interesting to see how well this has worked out. I'll switch gears to the third technology, microarrays. These require a little more explanation. So think of the glass slides like you might have had in high school biology. And there's all these little spots. If I blow up one spot, we'll see it's composed of many, many copies, up to maybe 1 million copies of a unique strand of DNA, in this case, ACGTs of DNA. So the idea here, I can have, depending on the type, tens of thousands, hundreds of thousands, up to several millions of these spots. And we pick what we're looking for, exactly what bit of DNA. So this is the idea behind the microarrays. And they can also analyze a complex environmental or clinical sample. We take the sample, we do an extraction of the DNA and RNA. Usually we do some kind of random amplification. We label it with fluorescence. This is actually what one of the glass slide type ones looks like. Gets scanned. This is the most expensive part. It's like a $125,000 fluorescent scanner. And then we do our data analysis. It's a cheap way to do the same thing you could do with sequencing. We'll talk about some of the trade-offs between the two. So we've developed a number of arrays. The one that's most pertinent to this discussion is called the LLMDA, Lawrence Livermore Microbial Detection Array. So we have probes on there for all sequenced microbial organisms. Every bacteria, virus, fungi, protozoa, archaea, and all of the parasites that have been sequenced as well. This kind of fits in a nice niche between PCR and sequencing. And this the yin-yang diagram here was to remind me to say that this is sort of the opposite, the inverse, if you will, of sequencing. With sequencing, you do nothing up front. You get millions to billions of reads of DNA, and then you spend a lot of time trying to map each read and figure out what it is. With the microarray, we looked at all the genomes first, analyzed them, compared them efficiently, and found which segments of DNA in each of the organisms best define it as unique from everything else. Then we put the probes for those onto the microarray. Our results then are very quick. We get our results in like 20 minutes off the microarray as opposed to hours off of sequencing, hours to days, depending on the power that you're using. So it's an interesting trade-off there. And it takes a lot of work to do this. This is one of those, don't try this at home, kids. It took about 15 CPU years the last time we redid the probe design. So we do this massive computation up front to know exactly what to look for, and then we stick it on the microarray and we go out and look for it. So the current version 7 now detects over 10,000 species, and you can see the split here in terms of how many different unique species. Sequences here is a fuzzy term. It can mean a genome. For viruses, it might mean a segment. Sometimes it's a split between the chromosomes and plasmids and so forth. It's a lot of data up there, and these numbers are going up quickly. And again, remember in 2000 when we started this, there were 62 in that box instead of the over 5,000 that we see now. Wow. Amazing amount of knowledge. And sometimes we build a vertebrate infecting only subset array. So we don't know in advance how these arrays are going to be used, but this looks for everything. So if you're worried about viruses that infect sea cucumbers, yeah, it's on here if somebody sequenced it. But if you're worried only about things that affect humans or dogs or animals that have commercial value, then we can use that array. We have been working with these in a huge range of things, and I'll just mention a few of these in detail in the next few slides. We've been doing some work on wound healing with colleagues at UC Davis and the Naval Medical Research Center. These are traumatic wounds from IEDs and other types of explosions. And with Veterans Day here, this is almost timely to talk about it. So we've been looking at profiling with both the array and sequencing, and people have also looked at some of the host biomarkers. The issue here is wound healing. Hopefully none of you have ever had a really bad wound, but all of us have had cuts, and sometimes the cuts heal up, you slap a Band-Aid, you forget about it, and other times they don't. And sometimes they get really bad and really infected. So what we're trying to figure out is, can we look at the microbes that are in the effluent, the pus or whatever that comes out of the wound, and can we predict whether it's going to heal or not? The interesting thing is, the answer is yes. And it's fairly bizarre. We found that there are certain profiles of types of bacteria. And ironically, if you have a higher concentration of gut-associated bacteria in your wound, you're probably going to heal better than if you don't. Nobody quite understands this yet. It's counterintuitive. It doesn't mean stop washing your hands. But there's some very interesting things that we're finding. So this is one of the types of areas that we work in. We also done some work in vaccine safety. short story here of Eric Delwer at San Francisco Blood Research Institute looked at the 8 vaccines that are given to young children. He was curious about, how, is there any contaminants in it? He gave us some blinded samples, we ran them, and we found that in one of the rotavirus ones, the rotavirus is there, and the log odds here is 550, and you probably have enough math to know, yeah, that's a pretty, you're really certain it's there, but we found at the same log odds score, there's something called porcine circovirus. When we first saw the output, and this is exactly what the output looks like coming out of our microarray analysis, we all said, porcine circovirus, what the heck is that? Well, it was just another one of those thousands of things that were in the database that we built probes for, and it turned out that in 2 years of clinical trials and I think 4 years of worldwide sale, there had been as much pig virus there as there was the rotavirus that was supposed to be there, and nobody knew it. It actually caused one of the most interesting moments in my career. In a span of 15 minutes after this story broke, the FDA halted the sales while they checked to see if there were any problems. Well, they'd had 6 years of giving it to kids, and there hadn't been a lot of babies dying, so there wasn't a problem. But one of my colleagues from the East Coast called up, and he was screaming at me, and he said, "You should not have done this study. He goes, you're a baby killer, Tom." And I, what? And he said, well, there's countries where this was the only rotavirus vaccine. This is an important disease in small children. It used to be 500,000 kids a year worldwide were dying of this. It's diarrhea. And this is a simple vaccine that can stop it. And so he's probably right. There was a few months where a couple kids didn't get it, but at least we're looking at it. Well, 15 minutes later, I got a call from a friend of mine and said, "Thank you for doing this work. My twins just had all those shots last week, and I'm glad somebody's out there trying to figure out how safe the things are we're putting in." So this is kind of the two sides of the coin that you find in this kind of business. Because of this work, we've been working closely the last few years with the CDC, and they have a new vaccine surveillance network. Turns out about 30% of the acute gastroenteritis cases are not diagnosed at all currently. It's just kids are sick, they don't know. And so our microarray and NextGen Sequencing are being analyzed by the CDC and their colleagues, trying to find better ways so that we can actually figure out what it is that is causing the disease better. We've been working with a lot of different people involved in cancers. This is just one example, people looking at bladder cancers. And the microarray found that there's a human herpesvirus 8 or Kaposi sarcoma-associated herpesvirus that is found in a large number of these bladder cancer samples. So this is yet another thing. There's a lot of work being done, and I think we'll see a tremendous amount happening just in the next couple of years. A lot of the easy cherry picking is going to happen, where people finding which viruses are associated with somehow causing or exacerbating various kinds of cancers. Very interesting work going on in many different kinds of tissues. The last sample here, and this is something that's really interesting. There's a great video on the web. I believe it's called "The Secret in the Bones," and Hendrik Poinar, P-O-I-N-A-R. It's really interesting. This guy is crazy. He goes out there and he finds old bodies. He takes the teeth, in this case from plague victims that were dug up in the UK. They knew from old records that this was a cemetery at the time of the Black Death. There was construction going on. They found some bodies. He got a hold of some of the teeth, and from the tooth pulp, they're able to extract DNA. Well, they'd spent 1.5 years trying to sequence it. It was just incredible labor. He then found out about our microarray, sent us some samples, and over 2 days, actually over Christmas a couple of years ago, we just stuck it on the microarray and got the immediate readout that it was Yersinia pestis, the causative agent of plague. The issue was that the DNA is so degraded after that many years. These tiny little pieces of DNA were maybe about 30 bases, very, very difficult to sequence, but a 30 base pair bit of DNA sticks just fine to our 60 base pair probes on the microarray. So now every scientific grave robber in the world is contacting us. We've done cholera victim, from cholera intestines that was in some lab somewhere. There's some people that have access to some 5,000-year-old bones from somewhere in the Sinai Desert, and we're trying to negotiate, seeing about that. In all these cases, what we're finding is these people almost certainly died of common diseases. Some people are trying to go a step further, and Hendrik is one of them, trying to actually sequence the entire genome of the plague from hundreds of years ago to try to look at how it has evolved differently. Really interesting stuff. All right, so let's go back to the whole thing here. So why isn't our doctor using these techniques. And my timeline is I got the idea for the microarray in 2003. It took a couple years to get funding. We've been testing it for 7 years. But the last time I had a bronchitis thing, I went in, the doctor listens to my chest. He goes, well, it's Friday afternoon, 4 o'clock. Writes me a prescription for some azithromycin to start taking these, and in 5 days you'll either feel better, you'll be in the ICU, or you'll be dead. You know, no idea what it was that caused it. I swear the next time I get bronchitis, I'm going to take a sample and run it on my array and come to my doctor and say, here's what it is. But the problem is you won't know how to use that information. So the issue here is having a good new technology is just the beginning of a very long journey. There's a lot of barriers, and this is something for those of you who want to be entrepreneurs, which I hope many of you are at this stage in your life. These are just things you ought to know about and be aware of. You might have a great idea of medical technology, but what does it take before you celebrate your first billion? It takes a lot. Right now, a couple problems. U.S. patent law, and I'm going to use arrays as my example. This is the nightmare I'm living. The patent law is murky on array probes. Yeah. The patent examiner, when he looked at our thing, which identified 10,000 things, and he said, describe 10 probes. 10 probes out of 500,000. So we picked 10, described them, and he spent a year, so he could throw a dart at each one of them. Here's some obscure journal that looks like it mentions the same DNA. Therefore, he threw them all out. It's crazy. The FDA, the Food and Drug Administration, doesn't know how to validate anything that big. So that's another problem. I've been working for 3 years very closely with the FDA, trying to help them along, and in the next couple of years, they'll figure that out. Here's something you might not have thought of. Insurance reimbursement for arrays is a problem, and I'll drill down on these slides. Finally, as I mentioned, the doctors don't yet know how to deal with the precise pathogen diagnostic. They weren't trained that way. And one for us computer folks, the hospital information systems can't handle the results. So there's a lot of problems, and I'll go down in a little more detail on each one of these. But the bottom line is the venture capital people, they kind of sort of know that there's these problems, and so therefore they're kind of skittish. So it's hard to get the funding you need to go in and really do something. So let's look at these. The patent law here is going on for years. It can't handle this massive parallelism. And the problem for us is you don't have to know the function of a region where we put a microarray probe to know that it's a good probe. We can determine that through comparative genomics. And the patent office has some problems with that. So this is a tough barrier. Well, I'm not trying to make this thing a commercial device. I'm not trying to make a whole lot of money off it. I'm trying to use it for other things. So I'm going to pass on that one. The FDA validation, the problem here is that a new device needs a gold standard comparison. Well, what is the gold standard comparison for an array that can detect everything? The only thing it could possibly be is sequencing. And sequencing hasn't yet been validated. So what's going to happen is we're getting close. In the next 2 or 3 years at most, sequencing will finally get validated as a diagnostic. And then microarrays are going to ride the coattails of that. Once that is there as a comparator, then it's going to be easy to show that, hey, a microarray can do this good of a job compared to sequencing. But I had to wait a long time for this to happen. So the insurance paradigm issue, one patient, one test, one result, one billing code. Very simple. That's how the insurances work. Well, arrays and other things break this. Now you have one patient, one test, many results. What do you do? So they're wrestling with that. Also, how do you bill for something you don't know to look for? So if you put a blood sample on an array and you're looking at sepsis, but you find out the person's got a couple of sexually transmitted diseases, but they didn't ask you about that. They're there for this infection problem. So there's some real problems with that, and they're wrestling with it. We've been told many times by people in the human health and also the food safety business, can you turn off everything that I don't think I need to know about right now? But can you turn it back on later when I'm required to? It's amazing. We talked to every Fortune 50 food producer, all the biggest ones, and I got a variant of that question from every one of them. So ultimately, the good news here, at some point, some of these insurers are going to get smart, and they're going to realize, hey, if we actually know what's making the people sick early, we can save money. And so there's going to be a paradigm shift, a big shift. I'm hoping that happens during this decade, and that's going to be really interesting when that happens. Then finally, the doctors are guessing if it's bacterial or viral. So when they've listened to my chest, I've actually had 2 times I've had some pneumonia, walking pneumonia-like thing since I've developed this microarray. And it might be viral, might be bacterial. When in doubt, hand out the antibiotics. So the current generation of doctors don't know how to use this. And the same is true for, they don't know for pathogen identification, or we could also identify the genes that cause certain antibiotic resistance. Don't give me Cipro. This particular bug that I've got has got Cipro resistance. The Cipro would be useless, but we don't have that capability now. These are things we're going to see happening fairly quickly. This is going to get resolved by time. So the young generation of doctors that are graduating now, maybe your older brothers and sisters in some cases, they're learning about genomics and they're going to be demanding this sort of knowledge. Finally, the thing about the hospital information system, that one patient, one test, one result, one billing code, that gets wired into databases, interfaces, and software in ways that this is about the only group I can ever talk to that would actually understand. When I say this to doctors or other researchers, they don't get it. They don't understand that somewhat the unwritten assumptions that we have in our software, how deeply that goes into the code. You may now, something that is one column in a table now needs to break out into a separate table with a one-to-many relationship joined between it. All the kinds of things that we understand as computer scientists. This is really tough. So there are changes being discussed that can handle all of this, microarrays and sequencing, the one test, multiple results. But everybody in the industry has to agree on those new standards before they can change. So this is the thing that's going on. It's ongoing. It'll happen this decade. And once it happens, it's, again, going to open the floodgate for all major changes in healthcare. So this thing, the bottom line for trying to get money to get a good idea, so the venture capital guys, they know that arrays, well, they're not really sure that arrays are going to compete with sequencing. I've had a good friend of mine for a few years been teasing me at conferences. He shows this sign of a melting snowman that says, "Soon I'm going to be dead," and he labels it microarrays. And I've been happy about that because my best competition has all gone into sequence analysis and has stayed away from arrays. But there's some interesting problems. So one of the issues here with sequencing is that it has the same problems with respect to arrays for clinical diagnosis. But right now, the focus on sequencing is human genomes for this precision medicine. Find the differences that each of you has that indicate whether or not you're more likely to have a certain cancer, certain disease, so forth. I think that the high throughput arrays that we hope to have available in the near future will cause some of the people to reconsider. I didn't show a slide, but I'm working with a company that is trying to get very, very high throughput arrays. So you could do like 3,000 samples in a 5-day, 8-hour work week workflow. How would you sequence 3,000 samples? You'd need a giant aircraft hangar full of sequencers. So sequencing currently inherently doesn't scale, at least not for pathogen diagnostics. I'll say a few things about personal health. This is not something I'm doing at Livermore. I'm working on the side with a couple of startup companies who are having a hard time getting VC funding. But it's amazing what you can do with your smartphones, all the kinds of analyses. You can get things to plug in and attach on there. But it's going to be a long path to integrate these into the health system. It's going to happen. It's already happening. There are health apps that are being sold, and the FDA is currently getting queasy about some of them because they appear to give diagnostic information without a real MD involved. So there's a interesting relationship there. Ultimately, I think it's going to be that these will tie in with your healthcare providers. So if you're under something like Kaiser or some HMO, that they're going to have apps and things for you to use. It's going to be very, very interesting to see how this goes. The vision of this really is that your healthcare provider is going to have some kind of information system cloud, and you're going to have things. So, when you get a little older, if you've got kids at 2 a.m., little junior screaming and sick, okay, your app is going to tell you plug this in, get the temperature, get a few other things. It's going to securely transmit the data back to the cloud, and then it's going to come back. And it's either going to tell you, give them children's Tylenol, bring them in tomorrow at 10 for an appointment, or rush to the emergency room, or whatever the appropriate diagnostic is. We will see more and more of these multiplex PCRs available in the various doctor's offices, and we'll see things, array-like things and sequencing-like things that are used. There's going to be this hierarchy. You start out with what you can tell quickly and cheaply, and you move it up as the case gets harder and harder to separate out. So my summary here is that medical diagnosis of pathogens is going to evolve extremely rapidly in this decade. It's just going to be really a lot of fun. We'll see these personal health applications blooming, and Ebola now is changing the landscape. I got an email right after I drove up here this morning. There's something like a $6 billion request to Congress for dealing with Ebola. A lot of that's going to, open the floodgates for better multiplex PCR. The arrays are going to come in sequencing. We're going to see a lot of uses, and we're going to see huge changes that are needed and are going to happen in all the things that I talked about. So the end tag here is, this is a great time for disruptive technologies. This is going to be a real interesting time for various kind of medical startups, diagnostic startups, all the kinds of things that are, don't go out and invent another startup that uses drones to deliver pizzas or burritos. I live in downtown San Francisco. There's at least 3 of those within 2 blocks of where I live. And, go out there and help us with medicine, please. So this is my colleagues on the team who extremely brilliant people, have done all the real work. And if any of you are curious to see about summer jobs at Livermore, go check. That's how I started my career, and it's been a fascinating ride since. I think I've got 5 minutes left for questions, so I'll be happy to take any questions.
Can you design an array that only detects things that would be a medical malpractice if not detected? That might motivate the insurers.
Yeah, it's hard to say. I mean, my feeling is identify everything and let people sort them out. So you can pick any list of things and you can identify. The same way you could have software that looked at metagenomic sequence data and only identified things on that list. Every attempt at list-based pathogen diagnostics that I've seen has failed spectacularly. I actually came up with the idea for this array in 2003 when I realized we were looking for a very short list of things in BioWatch. And what happens if you get hit with number 17 on somebody's top 10 list? You've got nothing. So it is an interesting thing. If we would never put porcine circovirus on an array to look at vaccines. Yes.
What was your background in biology before you started working at Lockheed?
I had 1 class in biology as a freshman undergraduate at University of San Francisco. So, I think the take-home message there is, as a computer scientist, you need to be nimble to adapt to specific domains. I've been almost all my career there, but I did work for a year in San Francisco in a management consulting, and I ended up becoming pretty good at construction, at database systems for large construction projects. I started my career helping to build the sewers of San Francisco, and it's been uphill since then.
Tom, I heard you mention archaea, but I don't think I heard you mention prions.
So good point. We get asked that a lot. Prions are misfolded proteins, and there is no detection capability that I'm aware of that can detect a misfolded protein, without going into somebody's brain. So no, nobody's got a good detection for prions, and I think we're very fortunate that hasn't become the huge problem that we thought 10, 15 years ago that it might be. But that is an open area, and we've no ideas even of how to do that in terms of a diagnostic that doesn't require you to have somebody's brain.
Yeah, there's a lot of precedent in medicine for finding things you didn't expect to find. So the concern you had about, you do the sample and you find all kinds of things you didn't know were there. For example, in radiology, I have a patient who came in with neck pain, and she had an X-ray, and they found a brain tumor because the radiology field was a little larger than it should have been. So they accidentally found a brain tumor. That's common all the time. Any physician will tell you that if you walk into UCSF with anything, you're going to diagnose 15 other things that are actually more dangerous than what you walked in with. So this kind of situation where you put out a sample, you get a lot of surprises. You have to decide clinically or some other way whether they're serious or not. Not so much whether they're billable, because we're all used to workarounds to figure out how to get paid, but it's more, what do you do with the information? And your comments about young physicians, I'm an old physician, and I've heard that forever, whether it's electronic medical records or anything. You graduate about 20,000 physicians a year, about 10,000 of them are reasonably bright. And there are 500,000 or 600,000 installed base. So replacing the old dumb ones like me with young smart ones doesn't happen fast. So putting hope in addition to the fact that the training for physicians has been dumbed down so much that your comment about they're not scientifically trained anymore, or now, they were much better scientifically trained 20, 30, 40 years ago. So I think those things you have to factor into the politics of it to have a realistic proposal. And it's like, sometimes the patients are really smart, smarter than an answering service, for example. And sometimes they're really stupid or very subjective about what's going on. So it's really exciting concepts you brought out here. That I think that the predictability of where to go, I understand why venture capitalists are tight with their wallets.
Points well taken.
It's really hard to figure out where to spend the money.
Thank you.
I have a quick question. I hope all of this is American. How about European or Asian?
That's really,
Yeah, so we certainly do a lot of work with people all over the world. U.S. is the only place that really is seriously worrying about the biothreat pathogens. UK to some much smaller degree, but a lot of places really aren't worrying about it as much. On the human health front, though, it's a lot easier. We actually have our microarray is being used in Denmark for patient analysis because they don't have the FDA. So, you know, if I get sick, I should go to Denmark and get analyzed, I guess, or something. So it's a very mixed bag all around, you know, all around the globe in terms of capabilities.
What's this about, are there problems with your microarray with respect to the size of the sample that is really carrying it? Thinking about aerosols, obviously, it's not easy to capture aerosols, then therefore will your microarray work?
Yeah, so the details actually of the limit of detection that we can do for the BioWatch, I can't give numbers, but a lot of work has gone into trying to get that as low as possible. Because, of course, your collector may be at the very fringe edge of a plume from where something was released. So a lot of work's done with that. We use random amplification in many cases to just amplify up everything that's in the sample. PCR and the microarrays with their specific detection can make use of that. One of the things I am working heavily with now is targeted amplification as a front end for DNA sequencing. There's a dirty little secret that if you take a current sequencer and you get your 24 million sequencing reads, even from a human sample, that's not a deep enough read to guarantee that you can get species level definition, much less strain or whether or not it's antibiotic resistant. So there's some really interesting things. You know, the emperor's clothes are not all that good in terms of sequencing. So watch that space. It's going to be a lot of fun next year.
Any other questions? Thank you very much.
Thank you.